Hi, everyone. I'd like to welcome Mike Webb to the conversation here. Mike, welcome. Good afternoon.
Thanks, Scott. Can you hear me okay?
We can hear you just fine. Thank you.
Fabulous.
Mike, do you want to give yourself a quick little brief bio about yourself and sell yourself up a little bit?
Okay, well, I spent the bulk of my career in GSK. I finished up heading up chemistry and analysis in the UK, but I did do a deep dive into oligos and worked in GSK to try and get our CMC response to. And David made a good point about picking these things up and dropping them too quickly, but we did involved in setting up oligo CMC and collaborations within the company. And like Chris Asahi, I've been consulting since about 2016, worked on double-stranded, single-stranded oligonucleotides, and I've now worked with most of the established CMOs, some big pharma, like Chris, some biotechs, with different flavors, and had some good collaborations with Ionis and Alnylam as well.
Great. Thanks, Mike. Thanks, and hopefully that this will shed some light on why the bullet points we're here to discuss why we're having you discuss these portions of it. So let's go ahead and just jump on into it. And can you shed some light, guide us on how to select the right partner to properly connect science and funding?
Yeah, that's a big subject, and we could do motherhood and apple pie because the right partnerships, any drug, right? But maybe the best way of approaching this is just, and David mentioned, and I think it's very important before we dive into rare diseases and genetic diseases, that we're moving into large things like dyslipidemia, Hep C, and that's going to require people to think about, with the CMOs, about large volume farming supply chains at the commercial end. And that's going to be a whole big partnership area that we're starting to think about now. But for now, let's think about where we are with small biotechs who are now pretty much modality agnostic, who are getting interested in oligonucleotides and maybe experts in a therapeutic area, but now are, like Chris was talking about, getting involved in oligonucleotides for the first time. Now, obviously, partnerships are important, but there's a few things that I think are important to recognize as special about oligonucleotides. As Chris said, oligonucleotides are real expensive to make and really expensive to develop, and the platforms are expensive. So for rare diseases, of course, they're less capitally intensive in terms of the clinic, in terms of the fact that there are fewer clinical centers, patient recruitment is easier, and patient numbers are smaller. So the cost of the CMC costs are quite high compared to the clinical costs, compared to, say, a conventional small molecule. So for a conventional small molecule, you're going fast to fail, either in the preclinical studies or in phase 1. With oligonucleotides, you already have a big attention from your funders about the partner make. So you need a CMO that you're going to work with. You need a CMO that understands its supply chain, and you need people like Chris and people like me. You need a drug product CMO that knows a little bit about oligos, and you need a drug substance CMO that may have to help you with the analysis of the drug product. That people recognize, outside of a DMF, the sponsor is responsible. So anything that you put into the patient, you as sponsor is responsible for. So your partnerships are incredibly important to your funders, and they're incredibly important to you. And we have a rapidly expanding CMO market. We have a lot of people out there, and as Chris said, they're all over the world. We're shipping oligos in cold chain all over the world. So setting up your partnership strategies are really, really important. And, we probably dig around with the other questions into this a little bit more about what are those important things. But the most important thing is your CMC is going to get the attention of your funders. So it would be the cheapest thing in the world would be to do what your tox batch and your first time in human clinical batch in one go. That's probably the most cost effective, but that's money up front, and that's going to get the attention of your investors. So you may want to back for your preclinical, and that has technical advantages because you can take a wider cut and purification, and you can have high levels of impurities qualified. So when you get into this as a new CMO, you need to understand the strategy, and you need to justify that strategy and costs. And you're really plotting that strategy with the end product in mind, because this isn't a med chem route you're going to scale up to a few kilos to go into phase 1. This is building the same basic process for the future. So those early partnerships, that CMO selection process that Chris just talked, is really, really important. And the connection between doing the right science at the right time and justifying this to the people who are funding you, be you in big pharma, and especially if you're in biotech, it requires a lot of thought. And that period between selecting your lead to go into your preclinical studies, at the point where you may be doing maximum repeated dose or maximum tolerated dose before you go into GLP, that period is a really important period to understand what you need to do in your strategy and get the funding to take you through first time in human.
It's great information, Mike. I appreciate the insight into it, because it's, again, some of it I don't even realize, or we don't have insight into that because we're not at that stage of the process. But building on what you said, though, as far as we're not only selecting the partnership that you're going to have with the CMOs, you also have to look at look who's going to agree upon developing, who's going to do that approach. And you mentioned here it's about front loading and who does the heavy lifting. Can you share decision ideas on that, and who does the decision making on that?
Yeah, so I think Chris alluded to that. You're going to put that heavy lifting into the hands of your CMO, or are you going to have an oversight with someone like Chris or someone like me they're going to help you with this? But I think it's important to reiterate the fact that with oligonucleotides, they're expensive to make. We typically make a more limited number of batches, particularly in rare diseases, than we do for volume diseases. So we typically, as Chris alluded, the solid state chemistry is basically the same. There are some differences, particularly between, if you're working with ion, big differences between the way ion do it and where a lot of the CMOs do it. But you are quite front loading your process, and you actually need that batch history. So it's really important to do the right amount of process development up front. And again, Chris alluded to this, so that you can start to build your knowledge base about your batches and your impurities from the get-go. So there is generally most consultants will say that you need to front load your process. The truth is, if you're a big pharma that's doing a collaboration, say with Ionis, you may only have one big batch, and then you're moving to a CMO quite late in the development cycle. And you may have to do quite a lot of work later on. And again, Chris mentioned the move between CMOs, and with new technologies and solution chemistry, again, which Chris alluded to, we may even be moving the process. So there is a decision to be made about where you're going to go with this process, what your doses are like, what your volumes like, what your market is like. And you really need to be thinking to having a target product profile at the beginning and deciding what you're going to do when. But in a lot of cases, particularly diseases, the best thing you can do is front load your process and build up a batch history, because you'll have a limited number of batches.
Which also then brings us into just manufacturing analysis challenges that you're alluding to. Can you shed some light on what you're currently seeing?
Yeah, so, I've been in this business a long time, and one of the early drugs that went on the market is amoxicillin. Amoxicillin is hygroscopic. It basically establishes water depending on the environment that it sits in. Oligonucleotides do the same. What I don't work out is why so much difficulty analyzing these materials when we've had them in our portfolio for 60 or 70 years, but we do. Assaying oligonucleotides with assaying oligonucleotides. The other problem is we don't have 99.9% pure sets, and we have separations that would make a small molecule chemist eyes bleed. So we have a lot of problems in assaying oligos. So the key question is, particularly for double-stranded oligos. So the particular, and there's a lot of literature out there, and I talk a lot about it at conferences in great detail. But there's correctly determining purity and correctly quantifying your impurities, and then correctly identifying your impurities and making sure that they're qualified when you go into tox. So the particularly double-stranded oligonucleotides and oligonucleotides with a lot of diversity of chemistry, you're going to get families of impurities, but you're going to get a lot of peaks in your chromatogram. And you're, particularly if you're using ion pairing methods, which most people do, and also anion exchange methods, you're going to have impurities that do not separate, depurination, oxidation, thioates. So it is very likely that you're going to be doing some work with quantitative mass spec, and therefore your QC methods, your analysis methods, and your ability to identify individual impurities is really difficult, really, really difficult. And then the other area, I feel a bit sad to come in the problems without necessarily having enough time to talk about what the potential solutions are. But then, that's how I get paid. Is you hire me and I'll help you. But the other thing that's kind of come across is if you take gamma ray ASOs, 20mers that are fully phosphothioated, and we do not control that introduction of the chiral sensor that thiolation produces. So your oligonucleotide is potentially a mixture of 500,000 compounds. There's been some literature suggesting that any variation in that diastereomeric ratio could affect the potency of your compound. And, the regulators are starting to ask questions of how reproducible is. Well, if you can get an analytical method to separate and quantify 500,000 compounds, I'd be really impressed. So we're having to look at that problem, semi-quantitative problems, or look at that problem during process optimization, process development, and process understanding, sort of things Chris was talking about. So those are the challenges, I think. Identifying the impurities, quantifying the impurities, determining purity correctly, so that you're meeting the standards of the guidances or the spirit of the guidances for synthetic small molecules, which at least, if not in detail, in spirit are applied to oligonucleotides. And so, Chris and his colleagues have done a phenomenal job in providing understanding for the synthesis. Analytically, we still have a lot of problems in giving you a really, really good, reliable number and a method that will work. So you're going to have a method that's got tiny shoulders and separations you're quantifying, and that method's got a life cycle of the product. So that's where I see a lot of the challenges.
Wow, it's more challenges than I would have. More challenges than I'm aware of. I'll put it that way, Mike.
Having said that, hats off to the instrument and column manufacturers. If we'd have tried to do this with the columns we had 20, 25 years ago, failed. We can do it now because these columns are so beautifully controlled.
Yeah, I agree with you on that. Let's touch a little bit on some of the supply side materials. And I know, Chris, we talked a little bit with him with the nitrile and the ideas and the short supply of it globally and how to deal with it. What are you seeing, and how do you guide your clients on how to forecast for their to support the clinical trials?
So, forecasting is difficult early on. You're still determining dose, but it's really important that you're able to forecast what your batch sizes are, and your CMO is able to forecast what to establish what is a reasonable yield and therefore a reasonable cost. Because in my former small molecule world, the drug substance was not a huge on the cost of therapy. For oligonucleotides, it's absolutely vital cost of therapy. The second thing is these batch sizes are quite significant in terms of supply and defensive stocks. And if you hold high defensive stocks, you have high working capital. But on the other hand, if it's for a rare disease, for a medically critical compound, you need expensive inventory. So figuring that out and figuring out that supply side, for a small biotech, and you want to go all the way to being a commercial supplier, you need to really understand that, because a stock out or a recall is the end. It's kind of the end. It may be the end of life for patients for rare diseases. So it's really important that's important. And then the other thing which I find a lot of my time talking about is it is okay to rely on your CMO to control your amidite supply and have quality agreements around amidite supply, but you're the sponsor. You are responsible. And so I think the whole of the pharma industry, and I hope big pharma colleagues will agree with me, haven't done a particularly good job in terms of change control upstream of their starting materials. Amidites are highly functionalized molecules, and we need to have supply agreements such that we know and we can evaluate any changes, because as you know, if you're adding a G 10 times at 0.1% impurity with the same coupling efficiency will give you a 1% impurity in your oligonucleotide. So a very small change in the impurity profile of your amidite has a very profound oligonucleotide. So you can rely on your CDMO to do that, but you sure as hell need to know that they understand the implications and they have good change control agreements with their suppliers. And that's really important, because those tiny changes could have a profound effect on quality, and that could lead to having a batch failure, which leads to a supply interruption. So this is a complex business, and this keeps guys like me in a job, guys like Chris in a job, and the platform experience sits with the CMOs and sits with us. And so when you're getting into this for the first time, Ionis have got have a platform, as Chris said, and so have R9. They've done a phenomenal job. But when you're getting it new, these are the major issues, in my opinion.
That's great. I'm glad you actually touched upon the ripple effect of having a impurity at that 10%. But if you're again loading it 10 times, as you mentioned, that's a 1% impurity already into your oligo chain by itself. And
yeah,
if again, depending on how you keep going the cycles, you do it maybe 10 more times, that number just keeps going, building. Your purity level keeps getting higher and higher. So that's very critical analysis that you have there. So we're getting too close to finishing up here. So Mike, let's actually roll into the last bullet point here of the growing role of CDMOs globally in choosing the partners to support bulk manufacturing. Big pharma, small biotech, choosing the right partner to support this. Can you shed some light on, or your thoughts on this topic?
Yeah, it seemed a couple of years ago, very capacity limited. Most of the big CDMOs have increased their capacity hugely. And also, we're seeing a lot of peptide manufacturers getting into oligos. I get a call maybe a couple of times a month saying, do you know any good oligo chemists? And I said, well, unless we clone Chris Oz, you may have to start with a synthetic chemist to learn this. So you really need to know that the people you're dealing with have the experience and the platform experience. One quick problem that we have is that platform experience is their know-how. That's their advantage. They work with a number of CMOs. Their platform experience they own, but it's difficult for them to share because they're under CDAs with their clients. So actually, guys like you and your organization, Scott and Cytiva, are really useful, because you have some of the knowledge that is very useful. Obviously, big guys like me and Chris especially can pick up that knowledge because we've done it many times. So it's really important for a biotech to to make sure they've got access to the knowledge that's going to do that early development that Chris talked about. And then the other thing is, with the advent of inclisiran, which David talked about, and hepatitis compounds, now oligos spill over in large volumes. We're seeing guide strands for CRISPR. These guys are seeing really big dollars coming through in having large volume. And if you're still looking at oligos for maybe, and even for oncology, which is very diverse, but also for rare diseases, you've got to make sure that you're going to get the attention of the really good guys in the CDMO who are going to be able to work on your lower volume, lower value compound. And, it's why, guys like me and Chris are around, which is that, there is a need for more people, more consultants to be able to help with that, because the amount of good process and analytical expertise is getting kind of diluted out, and there's some big manufacturing bucks. So for a biotech, their first oligo, there's the little bits of minefields around these collaborations that have got to be steered through so that they don't, end up, it doesn't end up going in the wrong direction. There are a lot of potential pitfalls. It's not like small molecules. It's not like biopharms. It has its own characteristics, but it is applied to the synthetic and analytical standards that exist in the regulatory world in the spirit of synthetic molecules. And there's still a lot we have to sort out in that direction.
Oh, great, Mike. Thanks for the insight there. I know for us, Asahi Kasei, as a company as a whole, we've been supporting this industry for well over 20 years. It's probably closer to 30 years, and 25-ish years probably, if I were actually pull the whole timeline back. And we're seeing the dramatic growth over the last 5-10 years of all the CMOs that are rapidly expanding, the number of CMOs that are coming online. But to us, what's even more exciting is to see the actual big pharma companies actually take on the manufacturing themselves and bring that back in-house to them. So while they don't necessarily have the technology or the insight to do that, they're quickly learning that. So I'm hoping that the industry itself is growing the knowledge and growing it's internally how they're going to be able to actually keep this manufacturing going, and they're learning by their own faults, and pluses that they're doing as well.
Yeah, and I would just add, I agree. In my world, what David said is incredibly important. That big pharma being in it for the long haul, either if they work with CDMOs or they, as Biogen are doing their own makes, especially that is going to help enormously. But it'll only help enormously if we move this forward together pre-competitively.
Yes, I completely agree. We need people to stick around the industry and keep pushing the industry as further along as we can. All right, Mike, thank you very much. I think that pretty much wraps up our conversations with the three experts here.